George grew up in California and went to UC Berkeley for undergrad. Then he went over to Delaware to get a master's in applied ecology, University of Delaware, and then he came back to California to UC Davis to do a PhD in Jay Rosenheim's lab. Now George is at Minnesota, where he's a professor, and he works primarily on classical and conservation biological control, usually with parasitoid wasps. And today he'll be speaking on the specificity and process of biological control using aphid parasitoids.
Thank you very much, Kate, for the introduction. Yeah, I think I'll need a little bit lower once the slides.
Yeah.
And I'd like to thank you all for coming at this odd time. Yeah, I'm not sure if you guys heard, but I was snowed in, which is what I get, I guess, for moving to the frigid north. It feels really warm to me out here, but Jay says it's like the coldest day of the year here, so that's just kind of how it is. But it's a pleasure to come back too. I think it's been 10 or so years since I've been back here. It's good to see some of the same faces here, but of course a lot of new ones too. And it's especially a pleasure to be able to talk about the work that I've been doing. So I titled this Specificity in the Process of Biological Control. So I think you'll see what I mean by that. I've been working mainly with soybeans and with parasitoids of the soybean aphid. And I'm not going to talk a lot about what the soybean aphid is. I think all that sort of we need to know for this discussion is that it's native to Asia, and it was first found in North America in the year 2000. It's basically spread throughout the soybean growing areas in the US and Canada, and is now the main agricultural pest for soy. And soybean didn't used to be sprayed, but now it is. And so I basically embarked in 2001 on a classical biocontrol project. Myself and colleagues went to Asia to look for parasitic wasps or parasitoids that attack soybean aphid. And this is just sort of a limited list of some of the species that we found during these studies. During these explorations. There are a number of species that are in the subfamily of Aphidiinae within the family Raconidae, which are all aphid parasitoids, and then also members of the genus Aphelinus in the family Aphilinidae, which are also all aphid parasitoids. I'm going to sort of talk about 5 of these species that we've brought in. Most of them, all of these have gone through quarantine post-specificity testing. And these 5, I think, sort of all tell different stories, and they all talk about sort of the interaction between host specificity or host range and the process of biological control. How we do biological control, what sorts of traits are either good or bad for biological control. So the first one is called Binodoxus communis. This is sort of the first one that we pushed through the pipeline. And I love this picture of this wasp because it shows how it stings. And a lot of people don't know that there are members of this group that have these claspers in the back of their abdomen that hold the aphid. So there are 2 claspers up here and 2 on the ventral side, and this is the ovipositor actually sticking the egg in. So there's a whole tribe of this subfamily that have this kind of oviposition behavior. That's not really the main point. The point is that we did these host specificity studies within quarantine. Basically, it's the safety testing that we now have to do in order to decide whether it makes sense to release a biological control agent. This is a kind of setup that we use. This is in quarantine. It's just a growth chamber showing these things that we call tubes, which have plants inside. Each plant had 50 aphids in our case, and then we put a single mated wasp into this arena. Came back 24 hours later, saw who was parasitized and who wasn't, and got sort of data. And in this data set, we have 20 aphid species that we tested. Some of these were on same host plant, some of them were on different host plants. 20 aphid species, and here what I'm showing is the number of adult female parasitoid offspring per one of those tubes. So, and here I've ranked the host use data. In this case, soybean aphid, Aphis glycines is on top, so that's good. But you can see there's this whole sort of range of other aphids. Some of them don't really appear to be hosts at all, or at least we don't get adult females. And then some of them have sort of intermediate levels. Is there a clicker, by the way? Is there a that's fine. Sorry. I'll just kind of keep walking back and forth. So one thing that we wanted to know is, in these cases of relatively poor hosts, why is that? Is it that they aren't getting eggs laid into them, or is it that there are eggs laid but they aren't developing? And I always tell my students that once you have a data set like this, it's like a gold mine, because every host has a different story. So for every host, there's a reason why it's not up here, why it's lower. And we didn't figure out the reason for each species, but we did for some of them. And I'm not going to go through the methods on everything that we did, because that would take a whole seminar, and I want to get to these other species too. But we did observations, and then we also did serial dissections of aphids that were stung, so that we're able to figure out whether sort of the limit was more behavioral or more physiological. So in a couple of species of aphids, eggs were never laid in those aphids, and we could tell through our observations that it was because the aphids were defending themselves. They were kicking, spinning around, pushing off with their antennae, doing all sorts of things, the result of which was an egg was never laid. So, you know, in this case we can say, you know, the reason, or at least, you know, really all that we can say is that these aphids never got eggs in them. You know, they might have been good aphid hosts, and we tried to get eggs into some of those. It's a long story. Some of them, another 4 species, were simply not detected by the wasp. So the wasp would walk over the aphid. You know, we had a protocol that the parasitoid's antennae had to touch the aphid for the aphid to fall into this category, or it was not detected. So the antenna touches the aphid, yet the parasitoid makes no motion to sting. So there's probably some sort of crypsis going on, where the parasitoid can't tell where it just walked on and touched this aphid. So these 6 aphid species never got eggs laid in them. Out of our initial set of 20 aphids, 9 of them did get stung, so did get eggs laid. And in those that were not soybean aphid, there's some level of physiological incompatibility. There's some reason that has to do with what's going on inside the body for why they're poorer hosts. 2 of them turn out to be host plant effects. So if you read the small print here, this is Aphis asclepiatus, which is a milkweed feeding aphid, and this is Aphis nerii, also a milkweed feeding aphid. When we raised these aphids on non-milkweed hosts, especially asclepiatus, came way up here. So it's clearly sort of a host plant mediated factor. With Aphis nerii, it's a bit more difficult. We couldn't actually rear it on a non-milkweed host plant, but we reared them on different species of milkweed, and that made a difference. In one of our aphid species, Aphis craxivora, a very poor host, it turned out that it harbors the endosymbiont Hamiltonella defensa, which you might know as the same bacterial endosymbiont which the pea aphid has and confers resistance to an aphidius herbi. So, and it's sort of a long story. The bacterium has a phage, and that phage produces a toxin which kills the eggs and or larvae of the parasitoid inside the aphid. So, in our initial study, we just found this pattern, and I'm not showing all the data, but the pattern was basically in our serial dissections that the eggs were dying and that the larvae were dying too, which is the same pattern that you find for the pea aphid. We subsequently did some studies where we cured some of them, and were able to prove that really Hamiltonella was the reason. So when we cure them, basically this line goes way up here again. So it's another case where we have some idea of why some of these hosts are suboptimal. In some of these other ones, there's a severe male bias sex ratio. So these are basically the tiny aphids. So in our behavioral assays, we always had them be the same size, but from our tube assays, we got these male bias sex ratios from small aphids. So that's another limiting factor. Something that you may have noticed, we can kind of go back here, actually. You can see that all of these almost are in the genus Aphis, and all of these almost are not. So there's clearly some sort of phylogenetic factor here. So just, another way that we can view these same data is, instead of ranking them, we can get a phylogeny of the aphids, which we did, and then you can sort of map these host parasitoid traits onto that. And so for this species, we basically find that the Aphis glycines clade, so the soybean aphid clade, is where you get the highest levels of parasitism. This is just shown a little bit differently. And so there's a way that you can assess the phylogenetic signal, and we use a program called Phylocom to do this. There's other ways, and we found that there was strong phylogenetic signal for the total number of mummies and the total number of adults. But you can see that even though there is phylogenetic signal to these traits, there's still really interesting things, like there's this species which is in this clade, but it does relatively poorly on that species. There's some of these that are sort of outliers that are relatively distantly related, but reasonably good hosts. So even though there's signal, there's still interesting things going on that aren't so tight. But this is supposed to be about biocontrol, right? We're supposed to be trying to determine whether we should release Phyllodoxis communis as a biological control agent of soybean aphid. All the stuff I just talked about doesn't really address that. What we need to do is think about what are the native non-target hosts. In our group of 20 aphids, there were actually only 5 that were native. Most of the aphids that we were working with were themselves introduced pests or just introduced non-pests. So these are the 5 non-targets. 2 of them were not hosts. 2 of them were relatively poor hosts. But one of them was actually a pretty good host. And this is called Aphis monardi. So this certainly gave us pause, We don't necessarily want to introduce a biological control agent that can have a negative effect on the native species, even if it's an aphid. Even the poor lowly aphids have a right to exist in their native ecosystems. So we wanted to look more closely at this one species, Aphis monarda. And I'm going to talk about some experiments that we did, but this one picture basically shows the whole story. So the sole host plant of Aphis monarda is Monarda fistulosa. There's also called bee balm. There's a bunch of common names for it, and it's a common native prairie plant in Minnesota and other areas. Do you have it around here? I don't remember it. Anyway, and this is the aphid right here. You can see that it likes to feed down in these flower racks, and you can also see it was tended by ants. We did a couple of things. The first thing is that we went out into the field, into these native prairie settings, and we found these flowers, and we tabulated how many aphids were on the flowers and whether they were ant-tended. And the first thing that we noticed is that the aphids tend to aggregate on the flowers. They're not on the leaves. All of our experiments in the lab were on leaves, on vegetative growth. It's very difficult in the greenhouse to grow these plants and to get them to bloom. So all of our experiments were on the leafy vegetative part of this plant, which seemed fine to us, but then we get into the field and we see, oh, the aphids aren't on the vegetative part, they're in the flower heads. So we did an experiment where we brought some flowers into the quarantine lab and looked at parasitism by Binodoxus communis on this non-target native aphid species, and we found that on the non-flowering vegetative growth, just as previously, we get a lot of mummies, but in the flowering bracts, if you give the aphids flowers, they'll settle in the flowers, and the parasitoids basically won't be able to find them nearly as well, and they don't get stung. The second thing we noticed in the field was that pretty much all of the aphid colonies were tended by ants. And in fact, colonies that were not tended by ants were very small, only 3 aphids. I don't know if you even call that a colony. So, they can only really live in colonies if they're tended by ants. So there are plenty of ants out there tending these aphids in prairie settings. And so we brought an ant colony into our quarantine lab, and we used Lasius neoniger, which was the most common ant that we found tending these. And we set up this kind of an arena. This is a box that has the ant colony that was dug up from the field, and then we just kind of connected our different arenas to the ant colonies in ways that we could compare plants that have ants, plants that don't. And this is on vegetative growth. And as previously, in cages without ants that have vegetative growth, you get plenty of mummies, but if you add the ants, much fewer. I think we all know what ants do with aphids. One of the things is that they protect aphids from natural enemies, and this shows it a little bit. You can see in this arena that this is a parasitoid, and she's actually got a sting. And this is an ant up here. I'm going to show a couple of pictures. You can see the ant moving down towards the parasitoid and just grabs it. Probably brings it back to its nest to feed to its larvae. And we also saw, I don't believe I have a picture, but we also saw a lot of the mummies were actually chewed, big hole in them. The ant had just come up to the mummy and taken the pupa of the parasitoid out. So based upon that information, on the laboratory host range studies and the sort of ecological host range studies on that one non-target aphid species, we got a permit to release Binodoxus communis in the field. And so we started to release it in 2007. And we did not get establishment, although we did find a few Binodoxus just this last field season. But we still have to figure out whether it's Binodoxus communis or the first record of a native North American Binodoxus species in Minnesota. So they may have established. Even if so, it established at a low rate. with great difficulty. And I'm not going to talk about this work, but we thought a lot about, well, why did it not establish or appear not to? And it does appear that it has trouble entering into diapause in the fall, which is unexpected since it comes from a part of China that has a climate very similar to Minnesota. You know, it's hard to imagine how you could even have genetic variability for the ability to diapause in a place like that. But maybe it did. There's a lot of intragular predation on this thing. So in Minnesota we have sources of intragular predation that we did not see in Asia, so it might be sort of out of whack in that way. And we also found that there was female bias dispersal from release sites. I'm not going to talk about these because it would take too long, and I want to get to these other species. But you know what I want to say for this one is that we found that it had a relatively narrow host range, that ecological risk was low, so we did a release, but the release didn't seem to work very well. I'll next talk about a species for which we still don't have a species name. So it's a species of Lipo-Alexis, and we brought this into the quarantine lab, and we did the same exact kind of studies that I showed you, but it had a much broader host range. First of all, and second of all, Aphis glycines wasn't even the best host. So we ran our phylocommon analysis on this, and there's no phylogenetic signal to these host parasitoid traits, which is sort of the hallmark of a generalist. Also, there's a host plant component here. The 5 best hosts, so are all grained aphids. So the common names here are bird cherry oat aphid, cornleaf aphid, green bug, English grain aphid, and Russian wheat aphid. And when we do a test of phylogenetic signal on plants, it's significant. But when we do a test of phylogenetic signal on aphids, it's not. So basically what Lipo-Alexis is a specialist on grain aphids, and it kind of does okay on the others. In terms of, is this going to be a promising agent for soybean aphid biological control? Not really. So, so biological control using this species, as I just said, it had a broad host range with a preference for grain aphids, so we didn't even pursue a permit for this one. Third species, Lysophlatus orientalis. This is one of our 3 species in this whole sort of odyssey. That's a new species to science. So this name is one that Peter Sari gave, as part of his work. I'll show you in a second that the host specificity is pretty high. This is also a rare member of this group, that's the Lyticus, so there are no males here, which from a biocontrol standpoint we kind of like. And we also, in the colonies, found a lot of winged mummies. And I'll show you a picture of a winged mummy in a second. What that basically means is that the parasitoids will lay an egg into an aphid, and then the aphid will sprout wings, fly away, or it could even be an adult aphid. So it could be a means by which the parasitoid is dispersed within the body of the aphid. So we kind of like that. So there are these 2 factors, actually these 3, which made it seem like a pretty promising agent. This is just a look at the host range. I don't have this sort of phylogenetically worked out yet. But it's a relatively narrow host range, basically on Aphis. Since there's only one sex, all we care about is weanlings, the mummies, and then the adults. Here's a picture of a winged mummy. But this story has a twist to it. It has what we think is a pretty interesting twist. And one thing that we were doing with all these species, or a lot of them, were what we call multi-generational cage studies. So these are the kinds of cages that we used in quarantine. Basically what we do is that we would seed the cages with 6 soy plants. Each of those plants had a certain number of aphids and a certain number of parasitoids. It was 50 aphids per plant and then 1 female per plant. And then we let them go. And every half week we'd take out 1 plant and we'd put in a new plant, but that new plant wouldn't have aphids on it. And this would go on for a couple of months, so that we're basically seeing the population dynamics that all started from that initial group. And for Binodoxus communis and for some of our other species, this is what we saw. So what I'm showing here, these are the weeks of the experiment of these multi-generational lab cage studies. What I'm showing up here is a cage that doesn't have any parasitoids in it, so a control cage. So the aphids get to about 1,000 or more per plant, which is basically their carrying capacity. Plants are killed at this stage, at this level, basically. And this line, though, is the number of aphids in cages that have Binodoxus communis in them, and these are the mummies. So you can see that when parasitoids are in the cages, the aphids kind of stay pretty high for a while, not very high. Then they go to extinction, as do the parasitoids. So that's exactly what we want to see. That's fine. This is part of the reason that we thought this would be a promising agent. So we did the same thing with Lysophlagus orientalis, and what we saw was kind of shocking. So first of all, here the control line is the same, but this other line that's really intertwined with the control cage line are the aphids that have Lysophlagus orientalis in the cage. You can see that there's there's no suppression going on at all. And it's not that the parasitoids didn't establish. They're there, and they're gaining in number, but they're not controlling the aphids at all. So, I mean, they're eating them, they're killing them, but it's not leading to lower population levels. So we looked at some other things too. One is the host phage preference. And there's already a little bit of a clue, because I previously said that we saw winged mummies, which suggests that later stages are stung. And indeed, so this is a comparison of Binodoxus communis, are these dark bars, and Lysophlagus are these light bars. These are the aphid stages, and this is sort of a preference, a relative preference for that stage. So for Binodoxus, you can see that there's a preference for the younger stages, and they don't really sting these, fourth they still do, but not these later stages as much as the earlier. Oh, I can say it here. As much as the earlier ones. Whereas Lysophlagus will basically sting any stage. So, including these stages that Binodoxus does not like. In addition, what we found was that when these later stages are stung, actually not only by Lysophlagus, but also by Binodoxus, those aphids are still able to reproduce, which makes sense. Like, think of an adult aphid being stung. These parasitoids are what we call coinobionts. So that means that the host doesn't immediately die when it's stung. It keeps feeding, even keeps growing, and it keeps reproducing. So these stages here, not the thirds. So the thirds will die prior to mummifying. But these later stages, if they're stung by Lysophlebus or orientalis, will spit out 6 to 10 nymphs, and then they'll die and mummify. You see what I mean? This actually isn't, the first time that this has been shown, but there aren't many parasitoids that sting these later stages. Mainly aphid parasitoids like these younger stages. So this could be contributing to the non-suppression. If you're stinging aphids and they're still reproducing, you're not killing them in the same way. Now, I should mention, though, that unstung nymphs will unstung aphids will produce about 10 times as many. So just by itself, this isn't going to get you to complete non-suppression. Something else, though, is that we followed those nymphs, so those 6 to 10 nymphs, and we looked at their fecundity versus regular old nymphs, or aphids. And so these here are the offspring of stung aphids, and you can see that they have a higher fecundity than unstung or regular old aphids. So these are the offspring of these stung aphids. Those 6 to 10 that they scored out prior to dying have a higher fitness. This does not translate into the second generation, which we needed to do because it could have been that these guys, the offspring of the stung aphids, were more fecund, but they're producing smaller or lower quality offspring. That's not the case because in the second generation there's no difference. So these guys, stung aphids, are laying nymphs that are simply more fit. And we actually have a theory for why that is. We pretty much feel like we have the mechanism nailed down, but I don't want to run out of time, so I'm going to go on. So, but we think that this might also contribute to the non-suppression. We have modeled this, and it doesn't get you all the way, so it can't actually explain all of the non-suppression that we found. So there's still more to learn, but it gets you some of the way there. So for biological control using Lysophlebus orientalis, so what have we done with this? So as I said, this is a promising species from the standpoint of safety. Pretty narrow host range, and then it has some of these traits which we like. But we did these experiments, and we found that there's some kind of compensation on the part of the aphids when they're stung, such that their offspring have higher fitness. There's no suppression in these cages, so we just decided, at least for now, not to go forward with trying to release this. It might be sort of very interesting case of a parasitoid making an aphid a worse pest than it was, or at least not having an effect on it. So, at least for now, we put the brakes on this one. Oh, this slide didn't come out very pretty. But we're on to the fourth species now, and we're now moving into a different taxonomic group of aphid parasitoids, members of the genus Aphelinus. So I'd like to tell you about Aphelinus certus first. And we did host range tests on this, very broad. Basically every single aphid that we gave it, except Russian reed aphid, who knows why, it's a great host. So, we just said, okay, this is one of those that's a generalist, and we don't want to go further with this. We didn't think about it very much. But then, starting in 2005, we started to see it in the field. So it basically became introduced on its own. Don't blame me. And So it was first found, let's see what I say in this slide. So it was first found in Pennsylvania, and then it was found in Canada. And if you talk to people studying soybean aphid in Ontario and Quebec, they'll tell you that this thing is single-handedly controlling soybean aphid. So it's like a natural experiment. And so of course, we were curious about whether it would get to Minnesota. It's been since found in Kentucky too. But we found it first in Minnesota in the summer of 2011. And in 2012, my student Joe Hazer did a survey of the state and basically found it in all of these places where there's a yellow dot. So it has sort of spread throughout the state. This is for 2012. And I want to share with you some studies that he did. Since it's a generalist, we were thinking about sort of apparent competition scenarios where maybe it's using other aphid species, maybe earlier in the year, maybe at the same time, and soybean aphid. And and then the question is, does that basically allow it to establish, does that lead to greater suppression on soybean aphid? One of the most obvious groups of other hosts of something like Aphids are the grain aphids. So when Joe did his survey, he found fields, soy fields that were either next to wheat fields or other soy fields. And this is actually a wheat field that's been harvested. He was timing his surveys when soybean aphids would be at their peak, which was actually when wheat is already harvested. And so, he was curious whether there's this kind of a link where sites that have had wheat have more Aphids on soy. And just to show you some of our beautiful landscapes in Minnesota, and how he was able to do this study, which is a natural study. It's so flat, and the landscape is basically corn and soybeans with some wheat sort of sprinkled in. So it was easy for him to find these comparison fields, paired sites that look basically identical, with the exception of the treatment that he wanted, which was adjacent to wheat or adjacent to soy. So this is a site where he would sample this soybean field and mark it as being next to a wheat field. And then here's a site that's soy next to soy, and they're just so similar. The road is the same, the tree. It's just so it's a nice study, I think, in terms of getting good pairs. And he did find that there was a higher density of Aphids certus per plant adjacent to wheat versus adjacent to soy. It's not a huge data set, and in fact, he did an experiment the following summer. Maybe some of you guys went to ESA, saw his talk, where he tested this in an experimental setting, and he could not replicate these results, so we're not sure. But I chose to show the cool looking ones rather than the negative data. But still, I think it makes sense to think of sort of a temporal apparent competition scenario. We like to call it the ghost of apparent competition past, where you have wheat as an earlier crop that has grain aphids that can harbor Aphid certus, and then those move on to soy. So what about biological control using Aphid certus? This is really unintended classical biological control. Nobody released this thing. It's basically an invasive species. It's very promising from an efficacy standpoint. But I think that the risk to non-target aphid is really high. And I predict that we're going to see negative consequences on native aphids. Lastly, that's not supposed to say 6, So this is a 5, there's only 5. I'd like you to meet Aphelinus glycinus. So this is one of our 3 species that's new to science too. Keith Hopper, who maybe many of you know, is my very close collaborator on this work, and he worked on both this one and on the host range studies for Aphelinus certus. So he just described this species as a new species, found that it has an extremely narrow host range. This truly is specialist of soybean aphid and coffee aphid. It really does very well on just those 2 species of everything that we've looked at. There's some other traits that we looked at. Remember, for Binodoxis, we were worried about the diapause issue. This one is very easy to get it to go into diapause using fall photo period, so we like that. So after a long time, because we had to put a name on it in order to get a permit for it, we had the data showing it was safe, way before we had the name, and you can't get a permit for something that doesn't have a name. But we did get a permit as of October 2012, and we started releases this year. So this is just a picture of one of our sites. This is the canisters that we use. So we're starting to release this. We have some data on the interactions between this and Aphelinus certus, in case you were wondering. And I have some slides on that, but I didn't put it in this talk because I know I'm running up against the time clock. Really, this one's just getting started. I personally view it as a biological control agent of soybean aphid, but also as a biological control agent of Aphelinus certus via the process of competition. I don't like that Aphelinus certus is here. I don't like the damage it's likely to have on native aphids. And we might be able to use Aphelinus glycinus to limit that damage. So to summarize, I've talked about 5 parasitoid species. Binodoxis communis, which is a specialist, and we did release it, but it seems like it didn't establish or so, not very well. Lycolexis was a generalist, and soybean aphid wasn't even its best host, so we did not pursue release on this one. Lysophlebus orientalis was a specialist, had some promising traits, but this bizarre novel compensation story that caused us pause. Aphelinus certus, a generalist that we did not want to release, but it came on its own. And then Aphelinus glycinus, a specialist that we're starting to work with in the field now. And I'd like to thank the people that work with me. Each species had sort of a group of students or postdocs that worked on it. And these are my funding sources. And I think it's almost 5 o'clock, so I'd better stop, and I'd like to thank you for your attention. If there are questions, I'd be glad to. Yes?
So I have a question about the permitting process. So you showed that for Binodoxis and Lysophlebus, they attack pretty much every aphis species that you looked at. In other words, how likely is it that some of the studies done in this kind of a system are missing some aphis species that might be even a better host, or how does that work with the permitting system? Are you required to sample extensively those species, or is it something that you don't really have to do?
Yeah. Well, Aphis is a big genus. It's the biggest genus in the Aphididae. It has about 400 species. So, yes, we certainly would have missed some native species. So our sampling of native species comes from going into prairies and finding the native aphid species that we can find. Some of those were aphids and some of them were not. So I think it's the kind of thing where sort of logistics kind of comes into play a little bit, because if you really are required to sample every native species out there, including really rare ones, then you basically can't do this kind of work. At the same time, the soybean crop is being sprayed with broad spectrum insecticide. So there's a little bit of not only risk assessment but risk analysis, where you're thinking about the risks of doing biological control versus the risks of not doing it. And we're always a little bit in the middle there. So yeah, does that answer your question? Okay. Mike?
I'm wondering, is Harmonia in this system?
Yeah. So when I said intragill predation, that's basically what it is. So Harmonia in the system is out of whack. There's way more here in North America than we see when we go to Asia. And so the intragill predation by that species is really high. It might be so high that it's really difficult for these guys to establish. One thing that we did show, though, is that Harmonia likes high densities of aphids. So there's less intragill predation at low densities of aphids. So as far as establishment, we sort of have a theory that it's better to establish the parasitoids at lower aphid densities. But yeah, I mean, it's a huge issue. But I also think of classical biological control of soybean aphid being classical biological control of Harmonia. So it's clear that so for those of you that don't know, Harmonia axaritis is the multicolored Asian lady beetle, and it's a coccinella that's considered to be highly invasive and is basically being implicated for the decline of native lady beetles, and its numbers ramped up really high once soybean aphid was introduced. So it's pretty clear that if soybean aphid levels go down, it's not like Harmonia will go locally extinct, but its numbers will go down too. So if we can achieve classical biological control of soybean aphid using these parasitoids, it should have a negative effect on the invasive Harmonia too. That's something that we'll ever know. Hugh, yeah?
Yeah, presumably there are also native parasitoids, right?
Right.
So that very first graph that you showed, supposing you had been using a native parasitoid, how would that have looked?
So the first graph I showed, you mean for the host range?
You had your soybean aphid was the most parasitized by your introduced parasite.
Yeah.
Right? Supposing you had a native, done exactly the same experiment.
Yeah.
Using a native parasitoid.
Yeah. So we have done that. So the most common native species in our area that attacks soybean aphid is Lysophlebus testacipes, which you also get here on cotton aphid. And it's sort of moved into the soybean system at a frustratingly slow pace. It does pretty well on soybean aphid, not better than its other hosts. It does great on Aphis maryi. It does great on. It's a very broad generalist. And soybean aphid is sort of middling. So, when we do phylogenetic signal studies of that for our host range, there's none. It's a generalist. So that one, it's really not any. It hasn't had a real impact. It's clear on soybean aphid numbers, and it's sort of a minor player. and I don't think there's a way to conserve
But
it.
that's in the last, the soybean aphid came in 10 years ago, is that right?
Yeah, 15
Okay.
years
Is there
ago.
any indication that it's evolving
Yeah.
as a better parasitoid?
Yeah, it's like we think it is, and then like there'll be some years where it does better than previous years. So I think, oh, that's happening. But then the following years it doesn't. So I think that its dynamics are very much tied to other aphids. So it's very well known as a green bug parasitoid in places like Oklahoma. And I think it's tied to things like green bug movement on wind currents from the south to the north. And it seems to be tied to Aphis cinereae, the milkweed aphis, So I think there's sort of outside forces. This thing, there's so much gene flow going on between Lysophlebus that attack certain hosts and other hosts that I don't think that it would evolve to be a specialist for soybean aphid very quickly. Yeah.
How long have the parasitoids been introduced and active in the field? How many seasons have
Oh,
gone through?
so Binodoxus was introduced in 1997. I'm sorry, in 2007. Apheline assert. I'm sorry.
How does it overwinter?
Yeah, well, we thought it didn't, and it still might not have. So, we did overwintering studies, and in those studies it did not. And a colleague of ours, Jacques Rodour, did some diapause studies, and he did show that there's, like way less than 1% did go into diapause, so there was some. In our studies it was none, but in his study there were like 1 or 2, but very low. So we thought that was a major limiting factor, that it wasn't able to overwinter. It did come from a part of China, the Heilongjiang region in the northeast. Is very cold. But we think, we have thought that it didn't. Then we found these 3 this last summer, which, it's really difficult to distinguish the species of that genus. But it's either Binodoxus communis or the first Minnesota record of a native Binodoxus. We just have to work through that.
Do you have any idea of what it does in China? How it overwinters there?
No. I've actually gone there to look at the overwintering host. And so we do so soybean aphid has an overwintering host and a summer host, and the overwintering host is buckthorn, so members of the genus Rhamnus. And so I have found parasitoids on Rhamnus in China, not that species, but Rhamnus is an overwintering host for both soybean aphid and cotton aphid in China, and so it's tricky. So we assume that it overwinters in the mummy stage. That's typical, and it might overwinter on the overwintering host of soybean aphid, or it might overwinter on a different host. This is not clear. Jay.
So I always wonder about how predictive the quarantine studies on parent rate but host range are going to be for field host specificity. So it seems like now you've got 3 species that vary in the level of specificity that you measured everything in the quarantine. They're out in the field now. Is there so do you think this is potentially an opportunity to see just how predictive the quarantine data are to natural conditions?
Oh, definitely for Apheline certus. So we're trying to get a handle on what it's doing in the field. So we've now reared it from soybean aphid, tertiary oat aphid, and English grain aphid. So, it is attacking the grain aphids. And for Aphelinus glycinus, if that one is established, we'll be able to do it. Binodoxus communis, if it did establish, then maybe we will. It's certainly possible that the host range in field is a lot narrower. That's sort of the common pattern. But, we like to sort of take a conservative approach and consider that there's risk to anything that can be attacked from the lab. Any Any other questions? Well, thanks a lot. Oh, Mike?
You mentioned you had a underlying theory for it, why the offspring would be.
Oh, yeah,
here.
yeah. So that underlying theory has to do with sort of the host-parasitoid interaction as it's known. So basically once one of these aphid parasitoids attacks a host, a couple of things happen. First, there's a venom, and that venom goes to the ovaries and it starts to degrade the ovarian tissue and also the young oocytes. You know, the purpose of that is basically to get the nutrients away from the aphid offspring so that the parasitoid offspring has access. So that's the first thing that happens. Second thing that happens is that when the egg hatches, when the parasitoid egg hatches, there's a membrane around that splits up into 32 cells called teratocytes. And these also migrate to the ovaries, and they tend to degrade the older eggs. And they also stimulate the primary endosymbionts, Buchnera, within aphids to produce more amino acids than they otherwise would. So basically, what's happening is that there are nutrients shunted to the shunted from oocytes and ovarian tissue to the parasitoid larva through the aphid hemolymph. So, you know, what we find, you know, what our basic theory is that those few aphid nymphs that make it are basically first of all, are free from competition because some of their siblings have died. So within the ovary. And second is that they're bathed in a nutrient bath, which is more nutritious than it would otherwise be. So, you know, we do find that the nymphs that are just about to be born, so within the ovaries, you know, we dissect aphids out. Those nymphs, prior to being born, are larger than regular nymphs from stung aphids. So it's sort of consistent with them being free from competition and or having more nutrients available during development. Does that make sense? Yes?
Yeah, so this may just be my sort of ignorance of like what happens with that. But I'm a little bit confused with like with Certus. So you were saying that you are releasing glycinus, yeah?
Yeah.
Well, might that have the potential to move Certus from attacking grain to other native aphids?
I think what's more likely, since soybean aphid is the dominant aphid, so numerically there's more soybean aphids and by biomass than any other aphids in this region. So I think the risk to non-target aphids is basically like a spillover, you know, either within season or spilling over and establishing. I think the lower that source is, the safer it would be to non-targets. That's my reasoning. You know, the scenario that you mentioned could happen too, I suppose. But I think, you know, you just have these huge blooms of aphids. And, you know, if you imagine, and, you know, we're seeing in 2013, I didn't show the data, but, you know, easily 50 aphilinosaurs per plant. And there's, you know, 150,000 soybean plants per acre and 7 million acres. So, you know, if we can reduce that sort of inoculum towards native aphids, you know, to me that seems like the best chance to reduce risk to non-targets. Does that make sense?
Yeah, I just didn't know, like, since it was single-handedly handling the problem in Canada, it maybe having some competition, like, you know, move them over to something else. I mean, since the one is a specialist in it, you know, I thought perhaps it would be quite effective also. Yeah.
I mean, I think what it, so we're now doing these sort of competition studies. Where we're pitting, you know, them sort of against each other.
Yeah.
You know, I don't have all the data yet. But I think what would happen is, so if Aphelinus glycinus outcompetes Sordus on a sort of per field or per plant basis, it would just lead to lower densities of Sordus in that area. So less of a chance for them to move on to into other settings.
Are the Canadians doing similar releases?
Pardon?
Are the Canadians doing similar releases to what you're doing?
No, the Canadians have not jumped on board for any of these other releases. They were interested in Binodoxus in the early days, but then they did those glyphosates in hand.
Can you speculate how Sordus actually got here?
Well, you know, I do think it's interesting because it was found on the East Coast. So it clearly, or, you know, it seems that it wasn't part of the original introduction of soybean aphid, which was first found in Wisconsin in 2000. So I think it's sort of a clue that there may have been a second introduction of soybean aphid to the East Coast a little bit later. Because it was first found in 2005 in Pennsylvania, and then found sort of in the general East Coast area. So, you know, I think either that happened, or it's been here much longer, maybe on a different aphid, maybe on grain aphids, and no one really noticed it, now it's moved on to soybean aphid. And it just happened to move on there first. Or, you know, it was there first. Any other questions? Thank you very much.
Thanks, George.
Parasitoids that attack soybean aphid. And this is just sort of a limited list of some of the species that we found during these studies, during these explorations. There are a number of species that are in the subfamily of Aphidiinae within the family Raconidae, which are all aphid parasitoids, and then also members of the genus Aphelinus in the family Aphilinidae, which are also all aphid parasitoids. I'm going to sort of talk about 5 of these species that we've brought in. Most of them, all of these have gone through quarantine post-specificity testing. And these 5, I think, sort of all tell different stories, and they all talk about sort of the interaction between host specificity or host range and the process of biological control. How we do biological control, what sorts of traits are either good or bad for biological control. So the first one is called Binodoxus communis. This is sort of the first one that we pushed through the pipeline. And I love this picture of this wasp because it shows how it stings. And a lot of people don't know that there are members of this group that have these claspers in the back of their abdomen that hold the aphid. So there are 2 claspers up here and 2 on the ventral side, and this is the ovipositor actually sticking the egg in. So there's a whole tribe of this subfamily that have this kind of oviposition behavior. That's not really the main point. The point is that we did these host specificity studies within quarantine. Basically, it's the safety testing that we now have to do in order to decide whether it makes sense to release a biological control agent. This is a kind of setup that we use. This is in quarantine. It's just a growth chamber showing these things that we call tubes, which have plants inside. Each plant had 50 aphids in our case, and then we put a single mated wasp into this arena. Came back 24 hours later, saw who was parasitized and who wasn't, and got sort of data. And in in this data set, we have 20 aphid species that we tested. Some of these were on same host plant, some of them were on different host plants. 20 aphid species, and here what I'm showing is the number of adult female parasitoid offspring per one of those tubes. So, and here I've ranked the host use data. In this case, soybean aphid, Aphis glycines is on top, so that's good. But you can see there's this whole sort of range of other aphids. Some of them don't really appear to be hosts at all, or at least we don't get adult females. And then some of them have sort of intermediate levels. Is there a clicker, by the way? Is there I'll just kind of keep going back. So one thing that we wanted to know is, in these cases of relatively poor hosts, why is that? Is it that they aren't getting eggs laid into them, or is it that there are eggs laid but they aren't developing? And I always tell my students that once you have a data set like this, it's like a gold mine, because every host has a different story. So for every host, there's a reason why it's not up here, why it's lower. And we didn't figure out the reason for each species, but we did for some of them. And I'm not going to go through the methods on everything that we did, because that would take a whole seminar, and I want to get to these other species too. But we did observations, and then we also did serial dissections of aphids that were stung, so that we're able to figure out whether sort of the limit was more behavioral or more physiological. So in a couple of species of aphids, eggs were never laid in those aphids, and we could tell through our observations that it was because the aphids were defending themselves. They were kicking, spinning around, pushing off with their antennae, doing all sorts of things, the result of which was an egg was never laid. So, in this case we can say, the reason, or at least, really all that we can say is that these aphids never got eggs in them. They might have been good aphid hosts, and we tried to get eggs into some of those. It's a long story. Some of them, another 4 species, were simply not detected by the wasp. So the wasp would walk over the aphid. We had a protocol that the parasitoid's antennae had to touch the aphid for the aphid to fall into this category, or it was not detected. So the antenna touches the aphid, yet the parasitoid makes no motion to sting. So there's probably some sort of crypsis going on where the parasitoid can't tell where it just walked on and touched as an aphid. So these 6 aphid species never got eggs laid in them. Out of our initial set of 20 aphids, 9 of them did get stung, so did get eggs laid. And in those that were not soybean aphid, there's some level of physiological incompatibility. There's some reason that has to do with what's going on inside the body for why they're poor hosts. 2 of them turn out to be host plant effects. So if you read the small print here, this is Aphis asclepiatus, which is a milkweed feeding aphid, and this is Aphis nerii, also a milkweed feeding aphid. When we raised these aphids on non-milkweed hosts, especially asclepiatus, came way up here. So it's clearly sort of a host plant mediated factor. With Aphis nerii, it's a bit more difficult. We couldn't actually rear it on a non-milkweed host plant, but we reared them on different species of milkweed, and that made a difference. In one of our aphid species, Aphis.
George grew up in California and went to UC Berkeley for undergrad. Then he went over to Delaware to get a master's in applied ecology. University of Delaware, and then he came back to California to UC Davis to do a PhD in Jay Rosenheim's lab. Now George is at Minnesota, where he's a professor, and he works primarily on classical and conservation biological control, usually with parasitoid wasps. And today he'll be speaking on the specificity and process of biological control using aphid parasitoids.
Thank you very much, Kate, for the introduction. Yeah, I think I'll need a little bit lower once the slides.
Yeah.
And I'd like to thank you all for coming at this odd time. Yeah, I'm not sure if you guys heard, but I was snowed in, which is what I get, I guess, for moving to the frigid north. It feels really warm to me out here, but Jay says it's like the coldest day of the year here, so that's just kind of how it is. But it's a pleasure to come back too. I think it's been 10 or so years since I've been back here. It's good to see some of the same faces here, but of course a lot of new ones too. And it's especially a pleasure to be able to talk about the work that I've been doing. So I titled this Specificity in the Process of Biological Control. So I think you'll see what I mean by that. I've been working mainly with soybeans and with parasitoids of the soybean aphid. And I'm not going to talk a lot about what the soybean aphid is. I think all that sort of we need to know for this discussion is that it's native to Asia, and it was first found in North America in the year 2000. It's basically spread throughout the soybean growing areas in the U.S. and Canada, and is now the main agricultural pest for soy. And soybean didn't used to be sprayed, but now it is. And so I basically embarked in 2001 on a classical biocontrol project. Myself and colleagues went to Asia to look for parasitic wasps or Parasivora, a very poor host. It turned out that it harbors the endosymbiont Hamiltonella defensa, which you might know as the same bacterial endosymbiont which the P. aphid has and confers resistance to Aphidius urbi. So, and it's sort of a long story. The bacterium has a phage, and that phage produces a toxin which kills the eggs and or larvae of the parasitoid inside the aphid. So, in our initial study, we just found this pattern, and I'm not showing all the data, but the pattern was basically in our serial dissections that the eggs were dying and that the larvae were dying too, which is the same pattern that you find for the P. aphid. We subsequently did some studies where we cured some of them, and were able to prove that really Hamiltonella was the reason. So when we cure them, basically this line goes way in here again. So it's another case where we have some idea of why some of these hosts are suboptimal. In some of these other ones, there's a severe male bias X ratio. So these are basically the tiny aphids. So in our behavioral assays, we always had them be the same size, but from our tube assays, we got these male bias sexifers from small aphids. So that's another limiting factor. Something that you may have noticed, we can kind of go back here, actually. You can see that all of these almost are homogeneous aphids, and all of these almost are not. So there's clearly some sort of phylogenetic factor here. So just, another way that we can view these same data is, instead of ranking them, we can get a phylogeny of the aphids, which we did, and then you can sort of map these host parasitoid traits onto that. And so for this species,